Right, good evening, people. Right, good evening, people. Please take paper number 6, paper 6. Mouse hidden. Mouse hidden. Right, people will discuss the MCQ paper. Paper number 6, MCQ paper. මේ Zoom එකේ මේ chat එක මෙහෙම යන එක නවත්තන්න බෑද? කරදරයක් ඒක. ඔක්කොම කම්පියුටර්ස් වල එහෙම යන්නේ. Right, people. දෙන්න. මේගොල්ලන්ට පේපර් එක පේනවද? මට පේන්නේ නැහැ. නේද? ස්ක්‍රීන් ෂෙයාරින් ගියාට දැන් මෙතන මගේ අර පොඩි වින්ඩෝ එකට එන්නේ. ඔව් හරි. Right, people, sorry about that. So, as usual, technical problems. Right, people, will check the MCQs first. Question number 1, consider following statements A and B. Consider following statements A and B. Right, people, first question. Consider following statements A and B. When temperature increase, the probability of collision between enzyme and the substrate increases. When the temperature increase, probability of collision between the enzyme and the substrate increase, correct me? Rate of enzyme catalyzed reaction increase with temperature until optimum temperature. Rate of enzyme catalyzed reaction increase with temperature until optimum temperature. Correct, neither. So they say that A correct, B wrong. A correct, B wrong. A wrong, B correct. A and B both wrong. A and B both correct. B is evident for A. Is that correct? temperature increase, probability of collision between the enzyme and the substrate increase, rate of enzyme catalyzed reaction increase with temperature until optimum. No, shall I take a fifth one as the answer? It does not, the explains the reason, isn't it? So I'm taking fifth one as the answer for the question. Question number 2, correct statement regarding enzymes. Correct statement regarding enzymes. Competitor inhibitors to the enzyme are competitor inhibitors to enzymes are irreversible. Competitive inhibitors are irreversible. Wrong, neither. They are reversible inhibitors. When an indicator binds with the active site, it stabilizes the inactive form of the enzyme. When an inhibitor binds with the active site, it stabilizes the inactive form of the enzyme. Wrong, neither. When the inhibitor binds, it stabilizes the inactive form. Then correct. Right? When an inhibitor binds with the active site, wrong. Neither. Active site is wrong. Ne? So it's wrong statement. You understood that? Fine. When an inhibitor bind with the active site, no, inhibitors don't bind with the active site. Inhibitors don't bind with, what they ask people? Correct statement. They ask for the correct statement. Huh? Competitive inhibitors, competitive inhibitors to the enzyme are irreversible. Wrong, ne? They want us to select the correct statement. Inhibitor bind with active site, wrong. ADP function as an allosteric inhibitor. ADP, activator, right? Wrong. Activators and inhibitors bind to regulatory sites. Where? Activators and inhibitors bind to regulatory sites. Where? Covalent interaction, no. To reverse from non-competitive, to reverse from the non-competitive inhibition, the substrate can be non-competitive inhibition. Substrate can be increased. Wrong, ne? I don't have any answer here. When an inhibitor bind with the active site is wrong, ne? Now, if there is no active site, this statement is okay. Right? So, all kiladaga one. All. No proper answer for that question. And if you have an answer, please let me know. Right? Question number 3. Which one of the following statement is correct regarding enzymes? Correct regarding enzymes. Some enzymes alter nature of the end products of the reaction. They are asking correct one. Wrong, ne? Inhibitors bind with the enzymes reversibly by covalent bonds. Inhibitors bind with enzymes. Inhibitors bind with enzymes reversibly by covalent bonds. This is correct, ne? Inhibitors bind reversibly, yeah, but then we have inhibitors. Irreversible inhibitors are present. Reversible inhibitors present. Inhibitors bind with enzyme reversible by covalent bonds. They are asking correct. So this statement doubtful, ne? Reversible inhibitors are there too. ATP act as an allosteric inhibitor to some enzymes. ATP act as allosteric inhibitor. ATP act as an allosteric inhibitor. Correct. Some non-competitive inhibitors bind with active site of the enzyme. Some non-competitive inhibitors bind with active site. Non-competitive, wrong, neither. Competitive bind with active site. The active site of an enzyme is made up of with many amino acid, wrong, neither. Few amino acid. So what's the best answer? I think you can go with the third one as the best. I'll take second also as wrong. Question number 4. Which one of the following statement is correct regarding factors affecting the rate of enzyme reaction? They are asking correct statement regarding factors affecting rate of enzyme action. Enzymes of different organisms have almost same optimum temperature. Enzymes of different organisms have almost same optimum temperature. Enzymes of different organisms have almost same optimum temperature. Wrong, neither. Normally, enzyme obtained temperature is around 37, but not for everyone. Not for everyone. So, you know, people, certain bacteria, they have optimum temperature around 70 to 80, 70, 80, little higher. So, components which are essential for the catalytic activities of certain enzymes are called cofactors. Components essential for the catalytic activity of certain enzymes are called cofactors. Enzyme cofactors. The rate of reaction increase until the enzyme molecule will be saturated at a particular enzyme concentration, substrate concentration. Most of the competitive inhibitors are irreversible inhibitors. Most of the competitive inhibitors are irreversible inhibitors. No. Wait. Most of the competitive inhibitors are irreversible, competitive nang reversible. Wrong. This is wrong. Rate of enzyme reaction increase proportionally with pH and then become constant. Enzyme reaction increase proportionately with pH and then become constant. Wrong. How about second one, people? Fourth one, second is okay. Second is okay. Moving to next page. Next set of questions. Question 5. Question 5. All the enzymes are globular proteins. All the enzymes are globular proteins. All enzymes are globular proteins. I will take it as correct, What they ask people here? We don't know, We'll check. Possess active sites. All enzymes possesses active sites. Correct. Increase activation energy. No, decreases, neither wrong. Used up in the reaction, no, wrong. Do not need cofactors which are non-proteins. No. All enzymes are globular proteins. All enzymes are globular proteins. All enzymes possess active sites. Possess active sites. Which one you think is correct? All enzymes are globular proteins. What was the statement given, people? Most enzymes are globular proteins. Because there are some enzymes, right? There are some enzymes, they are RNA molecules. Their RNA molecules can attach the enzymes too. Right? So I think we can go with the second as the answer. Second as the answer. What do you say about that? Right? What do you say about that? This says most of the enzymes are globular proteins, the resource book. Right? So we'll take people second one as the answer. Second. Question number 6. The above graph shows a rate of reaction versus substrate concentration. Rate of reaction versus substrate concentration. A, B, C, D. What is the correct combination of limiting factors in the graph for the alphabet A, B, C, D respectively? Right. What is A? A means people substrate limited. Neither. A, B. A is substrate concentration on x-axis. So therefore, this part substrate is limited. After this point, people substrate saturate. So B, no rate of change of rate of reaction because the enzymes are saturated with substrate. So A, enzyme, substrate concentration. B, A, substrate concentration. B, enzyme saturate with substrate. C, what is C, people? C is competitive. Ne? D is non-competitive inhibition. Non-competitive. So if you take that A, substrate concentration, increase of substrate concentration, let's say competitive C and D, substrate concentration, non-competitive, substrate concentration, competitive, non-competitive. Third is okay, ne? Substrate concentration, non-competitive and non-competitive. I think third makes a good description. B, maybe you can think of enzyme concentration because there is a fixed enzyme level, right? For the fixed enzyme level, you increase the substrate here up this part, right? So maybe the taking B as enzyme concentration, not wrong. So I'm taking third one as the answer. Hope you agree with me. Question number 7, people. Incorrect statement regarding enzymes. Incorrect statement regarding enzymes. Most are globular proteins. Globular proteins. Most are globular proteins, Harine. Catalyzed reactions are reversible. Hari. Some enzymes have sites other than active sites, correct. Most vitamins are coenzymes. Most enzymes possess only one subunit. How about taking fifth one as the answer? There can be several subunits in an enzyme. Question number 8, Carolus Linnaeus proposed a system of binomial nomenclature of species, which was accepted worldwide. Given below are scientific names of some species. Dipterocarpus zeylanicus, Homo sapiens, Coccus nucifera L, Pantherapardes scotia. Which of the above species name is accordance with Linnaeus? Dipterocarpus zeylanicus Hari. Homo sapiens, okay. Coccus nucifera L, this L is slanted, not correct. Neither. L shouldn't be italicized. Pantherapardes scotia. Scotia has to be italicized. So wrong. So AB must be the answer. AB is the answer. Right. Moving to next page. Moving to next page. Question number 9. Question number 9. A feature that can be seen only in two specific domains. A feature that can be seen only in two specific domains. Formylmethionine is the initiator, wrong. There are only one type of RNA out of RNA polymerase. Only one type of one type of our kinaka cut that ne. One type of RNA polymerase. One type of RNA polymerase only present in domain bacteria. No. Antibiotics inhibit growth. Antibiotics inhibit growth. No, not present in archaea and eukarya. Many genes have introns. Many genes have introns. Presence of histone protein associated. Many genes have introns, wrong, neither. Some genes, then presence of histone protein associated with DNA, that is correct. Fifth one is the best answer here. Question number 9. Fifth is the answer. Question number 10. Which of the following is unique feature of domain bacteria? Unique feature of domain bacteria. Membrane lipid is unbranched hydrocarbons. Membrane lipid is unbranched hydrocarbon. Membrane lipid is unbranched hydrocarbon. Feature of domain bacteria. Unbranched, correct. Prokaryotic, correct. Introns present in many genes, no. Formylmethylase initiator, correct. Histone associated DNA in some species, no. Unique feature of domain bacteria. Now, prokaryotic is correct, but it's not unique. Formylmethylase is the initiator amino acid, correct. Membrane lipid is unbranched, not unique. Why? Eukaryotes are unbranched too. So we have the answer now, fourth one. We have the answer, fourth. Question number 11, correct statement regarding history of classification. Aristotle classified plants according to lifespan. Aristotle, lifespan. Neither. Theophrastus, no. Theophrastus divided organisms into plants and animals, no. Ernest Haeckel classified organisms into kingdoms such as plant kingdom and animal kingdom. Ernest Haeckel. Domain classification based on mainly on the difference between prokaryotes. No. R.H. Whittaker not included prokaryotic and eukaryotic cell differentiation in 5 kingdom classification. Whittaker not included prokaryotic and eukaryotic cell differentiation, wrong. So can we take third one as the answer then? Third one as the answer. Am I wrong? Domain classification. Ernest Haeckel classified organisms into 2 kingdoms such as kingdom, plant kingdom and the animal kingdom. Neither. Aristotle identified plants and animals. That is what we said. Aristotle identified plants and animals. And then the Linnaeus identified plants and animals. Let me check that. He identified 2 kingdoms, plants and animals. So that's Linnaeus. Right. So I'll take this one as wrong. And then domain classification mainly based on difference between prokaryotes. Domain classification initiated like that. The key difference between the bacteria and eukarya. So I think we have to go for that one as the answer. Domain classification. Domain classification. They say people here, it became apparent every early evolution, some prokaryotes differ much from the others due from eukaryotes. Such difficulties have led biologists to adopt the 3-domain system. We'll take that as correct. We'll take it as correct. Fourth one is a better answer here. Right, question number 12. 3-domain classification system was introduced by 3-domain, Carl Voss. Question number 13. Which of the following statement correct regarding enzymes? Correct regarding enzymes. Biotin, one of the cofactor of enzyme. is coenzyme. Biotin. Biotin, one of the cofactor of enzyme, is coenzyme. Substrate largely change its shape due to active site. Wrong, neither. Enzymes are highly specific to substrate. Enzymes are highly specific. Substrate specificity. Enzymes are considered as polymers. Enzymes are polymers, correct, because they are proteins. So that means they are biological polymers. Some enzymes do not contain active sites. Wrong, neither. So A, C, D should be answer. Question number 14. Which of the following characteristic is common to domain archaea and bacteria? Prokaryotic cellular organization. Correct. Circular chromosomes. Correct. Presence of several kind of RNA polymerase. Wrong. You know, it's not present in bacteria but present in archaea. Sensitive to antibiotics. Bacteria but not present in archaea. Presence of heteroglycan in the cell wall. Bacteria but not in archaea. So AB must be the answer. Correct statement regarding history of classification. Correct statement regarding history of classification. Aristotle introduced kingdom Plantae and kingdom Animalia. No, that is actually Linnaeus, neither. Linnaeus classified plants according to lifespan. Wrong, neither. That is Theophrastus. The taxon phylum introduced by Ernest Haeckel. The highest level of taxonomic hierarchy was introduced by Carl Voss. The classification system introduced by Ernest Haeckel was based on nature of cellular organization. Classification system introduced Ernest Haeckel was based on the nature of cellular organization. Wrong, ne? So we'll take CD as the answer. CD as the answer. Right, moving to structured part, people, I have given you the marking scheme. Right, I have given you the marking scheme, and then the marking scheme is in the chat line also. Right, so we'll quickly go through that. Right, what is an enzyme? Check the marking scheme, okay, people? I'm not going to write it today. What is an enzyme? Macromolecule which act as biological catalyst, produce inside living cells. Give three general characteristics of enzymes common to all enzymes. Biological catalyst. Their presence do not alter the nature of the properties of the end products. Enzymes are highly specific, substrate specific, not being used up during reactions. Then they possess active sites where reaction take place. What are known as active sites of an enzyme? Specific region on the enzyme to which the substrate bind. Specific region of the enzymes to which the substrate bind. Right. Briefly explain the induced fit hypothesis of enzymatic reaction. Induced fit. Enzymes are not rigid structures. The interaction between the substrate and the active site may slightly change the shape of the active site so that the substrate and the active site become complementary to each other, and this makes a better fit. Ne? Those points you write here. I'm not going to write that, people. I think you have, it's not necessary to write. No need. What are cofactors, people? Non-proteinous components which are essential for the catalytic activity. They are the cofactors. And then indicate two examples for organic and inorganic cofactors. Organic cofactors, what are you going to take, people? NAD, NAD plus NADP, FAD, biotin, FAD, biotin, derivatives of vitamin. Inorganic, Zn plus 2, Fe plus 2, and Cu plus 2. Give another name for organic cofactors. What do you call them, people? Coenzymes, ne? Coenzymes. Coenzymes. Right, part B1. Part B1. Name factors that affect the rate of enzyme reaction. Temperature, pH, substrate concentration, inhibitors, substrate concentration, inhibitors, etc. Right, moving to next page. What is the temperature at which the maximum rate of enzyme reaction observed is called? What do you call that, people? Optimum temperature. Optimum temperature. What is the reason for the rapid decline of the enzyme activity when the temperature increases beyond the optimum temperature? What are you going to write, people? Hydrogen bonds, ionic bonds, and other weak interactions break. Right? This results change of the active site, which alters the complementary binding of the enzyme and the substrate. Right? Check that given answer. The hydrogen bonds, ionic bonds, and other weak interactions break with increasing of temperature above the optimum. This results in the change in the active site of the enzyme and complementary binding of the substrate and the enzyme. What is the above event called? What do you call, people? Denaturation of enzymes. Heat denaturation. Heat denaturation. What is known as optimum pH? The narrow range. Optimum pH. The narrow range of the pH which a particular enzyme catalyzed reaction. We'll say that, people, the pH at which the highest rate of reaction occur. pH at which the highest rate of reaction occur. Give optimum pH for following: trypsin, what we said, trypsin 8, pepsin 2, pepsin 2. Is that given? Check that enzyme. It was given 6 to 8 kilo pH 6 to 8 for most enzymes. Pepsin works trypsin 8, pepsin 2. Trypsin 8, pepsin 2. Right, next one, people, what are inhibitors? Certain molecules or ions selectively bind permanently or temporarily to the enzyme molecule and prevent them forming enzyme substrate complex. Certain molecules or ions selectively bind permanently or temporarily to the enzyme, forming to prevent them forming enzyme substrate complex. Mention 2 types of inhibitors and give an example for each. 2 types of inhibitors: irreversible inhibitors. The other one is reversible. What are you going to write as irreversible, people? Toxin, poison. Reversible, drugs used against microbial infections. Give two differences between non-competitive and competitive inhibitors. Two differences between non-competitive and competitive. This side is non-competitive. This side is competitive. Then non-competitive do not compete with the substrate. Competitive compete with the substrate. Non-competitive bind to site other than the active site. Competitive bind to active site. Non-competitive inhibition cannot overcome by increasing substrate concentration. Competitive inhibition can overcome by increasing substrate concentration. Check the marking scheme. C1, name three types of regulation mechanisms of enzyme, allosteric regulator and inhibitors, cooperativity and feedback inhibition. Take the role of the following substrate substances in the allosteric regulation. ADP. What are you going to say, people? Act as an allosteric activator. ATP, act as allosteric inhibitor. What is the significance of feedback inhibition? What is the significance of feedback inhibition? What we said, people, end product goes an effect on one of the enzyme involving the pathway to reduce its action. Prevent wastage of chemical, regulate and end products produce metabolism. Regulate end products produce in metabolism. That is the given one. Significance of feedback inhibition. So we'll take the scheme answer. What is that, people? Take that prevent wastage of chemical resources. Give an example of feedback inhibition. Feedback inhibition, ATP, when supply exceeds the demand, ATP itself slow down the ATP production. ATP slow down the ATP production. Right. Next one, people, second question. What is meant by classification? Arrangement of organisms into groups for convenience of studying. Arrangement of groups on the basis of common characters. What are changes that can be observed on following criteria hierarchy of taxa from domain to species? Domain to species, number of individuals decreases, decrease. Number of common characters increase. Common characters increase. Common characters increase. Next one. What is the scientific name of horror plant, which is indigenous? Dipterocarpus silenicus. What is the important rule acquired to follow when handwritten of scientific name? What is the important rule required to follow when handwritten? Underline the name. Underline each part of the name separately. Write two criteria used by Carolus Linnaeus for classification of flowering plants. What we said, people, floral features, number of stamens, number of styles of the flower. Number of stamens and number of styles of the flowers. What is biological definition of a species? Biological definition of a species. Group of organisms share many characters in common that has the ability to interbreed and produce fertile offsprings. Next one. Give phylogenetic species concept. Give phylogenetic species concept. What we said, people, in phylogenetic species concept? We said smallest group of individuals that share common ancestor. Smallest group of individuals that share common ancestor. Name the domain eukarya. Name the domain eukarya into which only multicellular organisms are included. Into which only multicellular organisms are included. Domain eukarya into which only multicellular organisms are included. Plantae, Animalia, right? Plantae and Animalia. Only multicellular organisms are included. I think in the marking scheme it's given as domain eukarya, wrong, ne? What are you going to write, people? Name, they're asking domain name. They're asking domain name, not the kingdom name. So then people eukarya, domain eukarya, right? Name two indicators used by Aristotle when classifying organisms. Indicators used by Aristotle when classifying organisms. What we said, people? Mode of nutrition, sorry, mode of locomotion, reproduction, presence, absence of red blood cells. Name the kingdoms which microorganism belongs according to the classification introduced by Whitaker. Name the kingdoms to which microorganisms belongs according to the classification. What are you going to write, people? Fungi, Protista, Monera. Kingdom Monera, ne? Write scientific name a plant species endemic to Sri Lanka. You know that. Dipterocarpus silenicus. Write the scientific name of, okay. Write the basis of present system of classification. Sequence of bases of DNA of important, sequence of bases of DNA of important genes. The sequence of DNA of mitochondrial and chloroplast DNA. Sequence of bases of ribosomal RNA. Molecular structure of cellular components. Amino acid sequence of important proteins. Right. Part C1, write advantage of artificial classification. Advantage of artificial, easy to use and expand by adding more groups. State the difference between artificial classification and natural classification. Artificial classification based on few characters, natural classification based on many characters. Artificial classification, evolution relationships are ignored, evolution relationships are considered. Only system used before 18th century, artificial use after acceptance of the Darwin's theory of evolution. Or used after 18th century. Ne? State international codes of binomial nomenclature. Two species of organisms cannot have the same name. Each species should consist of a generic name and a specific epithet. Name should be made up of Latinized words and written in Roman scripts. Should be underlined when handwritten. The first letter of the genus name must be capitalized. Right, next one. State two organisms, groups which considered as two separate artificial groups. One is people virus and protista. They say two groups of organisms, virus and protista. State the name of the scientist who introduced the following taxon. Phylum, Ernest Haeckel. Domain, Carolus Linnaeus. Sorry, Carl Voss. Kingdom fungi. Kingdom fungi, Robert H. Whitaker. Taxon, order, Carolus Linnaeus. Which criteria used Robert H. Whitaker in his classification system? What we said, people, cellular level organization, then unicellular, multicellular nature, nutrition, right? Mode of nutrition. Right, then people, two essay questions are there. So let's check that marking scheme given for the essay question. Regat pada balan lamai. Right. What is the question? Explain the mechanism of enzyme action in living cells. Explain the mechanism of enzyme action. Mechanism neither. So what are you going to write here, people? Mechanism of enzyme action. Enzyme and the substrate form enzyme substrate complex. The catalytic action of the substrate catalytic action of the enzyme convert substrate into product. The enzymes are specific, and then the region is the active site. It has few amino acid. The shape of the active site is complementary, but always not fully complementary. So therefore, enzymes are not rigid. They can slightly change the shape of the active site. That makes a perfect fit with the substrate. So once the enzyme substrate complex form the product, then they depart from the enzyme. Next one, we will describe the allosteric activation and inhibition procedure in the regulating mechanism of enzyme activity in cells. Allosteric activation and inhibition. So there is a whole paragraph for that. Right? You have to say that the enzymes, they oscillate between two forms, the active form and the inactive form. So when the regulatory molecules, they bind to a site other than the active site is called the allosteric site. When activator binds, stabilizes the actives, the conformation. And then so more products can be formed, people, by this interaction, right? And same like that, when the inhibitor bind, inactivate the enzyme. Example, ADP function as allosteric activator. Right? So when ATP supply exceed the demand, sorry, ATP act as allosteric inhibitor, exceed the demand, it inhibits the enzyme action. Next one, people write short notes, history of classification. So this is what I have given, right? Following the resource book, you have to write the summary. Binomial nomenclature, you have to write the rules of the binomial nomenclature. I think those things you can write, and those who have submit the paper, right? Characteristic features of bacteria, right? Those who have submit the paper will, I am sorry, I didn't notice that. I didn't notice it. I didn't share it, right? So forgot to share that. So this part, all right. So hope you have submitted this one. So submit, we'll correct them and hand over back to you. But now it's too late for paper 6. Why don't you try from paper 7? Paper 7, submit and give it. Right, I'll give you paper 7 on coming Saturday. Right, then thank you very much for your time and your participation. I'll see you all soon. Right, so have a nice time and bye bye. See you.